Welcome to Advalytics, Olympus's worldwide competence center for cell-based molecular biology. It is our mission to make single-cell analyses simple, seamless, and sensitive. At the heart of our single-cell analyses platform are the AmpliGrid slide and the AmpliSpeed slide cycler. The AmpliSpeed comes configured to thermocycle 1, 2, or 4 AmpliGrid slides simultaneously. The AmpliGrid has 48 reaction sites. As you can see in the cross-section, the reaction takes place in a hydrophilic anchor region that is surrounded by a hydrophobic ring. The hydrophobic ring ensures that the 1 microliter reaction mix stays in place. We cover the reaction mix with oil to prevent evaporation during cycling. Working in precisely defined 1 microliter reaction volumes on an inert glass substrate is the secret behind the amazing sensitivity of the AmpliGrid platform. In order to harness the single-cell sensitivity of our platform, we have developed a simple and seamless 4-step workflow. You can use flow cytometry, laser capture, or micromanipulation to deposit cells onto the AmpliGrid slide. Then you can optically verify that the cell is placed correctly, a feat that is impossible if you use tubes or microtiter plates to hold your single cells. Now you run your molecular reaction on our AmpliSpeed slide cycler. Finally, you analyze results with the method of your choice: gel, capillary electrophoresis, sequencing, or qPCR. Now let me show you how this all works. Let's start with cell deposition via flow cytometry. If you are sorting with a Becton-Dickinson fax aria, simply place the AmpliGrid into our fax adapter and insert the adapter into the aria's sorting bay. Using the aria's Diva software, it is easy to define the AmpliGrid as a device, specify single-cell deposition, and monitor progress. The close-up shows how single cells, embedded in tiny drops of sheath fluid, are deposited onto the AmpliGrid. After the sort is completed, remove the slide from the bay. Alternatively, you can use micromanipulation to deposit cells on the AmpliGrid. A typical micromanipulator setup consists of an inverted microscope such as the Olympus iX61 shown here and a joystick-operated micromanipulation capillary. Place an AmpliGrid slide next to your sample in the optical path of the microscope. Using the control knobs, you move the capillary into your sample. Under the screen, you locate a cell of interest and aspirate the cell into the capillary. Retract the capillary and move the microscope stage to position the capillary on top of your target AmpliGrid site. When the capillary is directly over your chosen AmpliGrid site, you eject the cell while visually monitoring successful transfer through the microscope. This is a key advantage of the AmpliGrid platform. The ability to verify correct cell placement prior to molecular analyses is a key advantage of the AmpliGrid platform. Simply insert the AmpliGrid slide into a fluorescence microscope. Scan the slide and focus in on the cells in the reaction centers, a process for which automated solutions are available. Finally, store your quality control results. Now we are ready to use the world's smallest thermocycler, the AmpliSpeed, to amplify the DNA, messenger RNA, or mtDNA directly from the deposited cells. First, we pipette 1 microliter assays, shown in red, on top of the cells that we deposited in the reaction sites. Even if your positioning is slightly off, the hydrophobic ring surrounding the reaction site forces the assay right where it belongs. Pipetting 1 microliter volumes has never been this easy. Next, we cover the assays with 5 microliter of sealing solution so the assay does not evaporate during amplification. Of course, pipetting can be automated. Here you see the Eppendorf EP motion system automatically pipetting assay and sealing solution onto AmpliGrid. Using an intuitive graphic user interface, it is quick to enter any cycling program into the AmpliSpeed. Insert the loaded AmpliGrid, close the lid, and start your cycling program. The AmpliSpeed does not use Peltier elements, but rather transfers heat with a built-in thermal mass. That is the secret behind AmpliSpeed's small size, whisper-quiet operation, low heat emissions, and extremely low power consumption. After amplification, we are ready for analyses with the method of your choice: gel, capillary electrophoresis, sequencing, or qPCR. Once the cycling program is completed, take the slide out of the cycler. If your research calls for gel analyses, add loading dye, here colored in green for better visualization. Gently touch the top of the sealing solution with a drop of loading dye at the tip of your pipette. It will detach, tunnel through the sealing solution, and mix with the amplicon. Next, we remove the mixture from the slide and transfer it into the gel. Pierce the pipette tip through the sealing solution and gently aspirate the mixture. Using a Cambrax flash gel, results become visible after only a few minutes. The outside lanes contain the reference ladders. The middle lanes each represent the results of one single cell. Here we used an assay for 3 housekeeping genes. For each cell, all 3 bands are clearly visible. Proof of the AmpliGrid system's single-cell sensitivity. You can also automate retrieval and transfer of amplicons into microtiter plates for analyses by capillary electrophoresis or qPCR. Here you see the Eppendorf EP motion retrieving amplicons and pipetting them into microtiter plates.